Welcome back from the procedure. I hope everyone had fun watching how kombucha was made. So we're going to enter the results section at this point. And for the results, you're not actually going to be able to honestly answer these questions if I'm being honest. So because we're in a remote situation this year, sadly, we weren't able to make the kombucha as a class and actually have you taste it. If you are feeling extra, I very much invite you to go to a grocery store and just buy yourself a bottle of it just to try it. If you're curious, you don't have to, obviously. So if you do that, answer these questions. Was your kombucha fizzy and did your kombucha taste fermented? And you'll be like, it's one of those tastes when you taste it. But if you didn't get a chance to try the kombucha, you can just type, I did not try it for these questions. That is perfectly okay. All right, so let's move on to the conclusion section. So this will actually be asking us about the science behind this fermentation and kombucha brewing process. All right, so this says here, we're going to be answering these questions essentially for the conclusion. It says the reactants of cellular respiration are glucose, which is sugar, and oxygen, O2. How did we supply these reactants to the kombucha? So your job is to answer these questions. Where did we get the sugar from? So sugar was added directly to the tea after it had been brewed, and then oxygen was added to the kombucha because of the cloth covering the jar. What can we say? Airflow, or I can say air was able to flow. Air was able to flow in and out of the jar during the first fermentation process. All right, so did I spell kombucha wrong? Come on, computer. There we go. Okay. So you do not have to write what I write word for word. Just get the gist of it, please. So let's move on to the next question. The products of cellular respiration are carbon dioxide and water. This is what we are creating. Where can we see both of these products in the kombucha? So we can see the carbon dioxide in the fizziness of the drink. We can see the water in the fact that this is a liquid that we drink. So there's going to be water because it's a liquid. Oh, I forgot the I. Liquid. All right. Moving on to number 3. While yeast requires oxygen for cellular respiration, bacteria do not. They respire anaerobically and make ethanol alcohol as a product. Where can we see this product in the kombucha? So we can see the how do I spell ethanol? Ethanol alcohol in the kombucha. Alcohol. There we go. How am I not spelling this right? All right, we can see ethanol alcohol in the kombucha through the flavor of the drink. So if you do end up trying this out for yourself, again, you will get that like flavor, and that would be the alcohol. Very little amount, again, completely legal for you to buy this and drink this. It is a very, very low amount. It is sold in grocery stores. You do not need an ID to buy it, but you can taste that like puckering taste. All right, so will you continue drinking kombucha after this lab, or at least try it? Why or why not? That is up to you. So if you did try it, did you like it? If you didn't get a chance to try it, are you now interested in trying it out? just give me a heads up. Let me know if I got you intrigued by kombucha. And then for the last question, what do you think the SCOBY feels like if you touch it? Would you be willing to poke one? So again, that is up to you. You let me know if you feel like getting your hands dirty with the SCOBY, but let me know. I'm just curious. So that would be your conclusion. And then we're going to finish up with our experimental variables over here. So let's take a look. The control for this lab would be the starter fluid of the kombucha. So the kombucha I made from my last brew of it. That's the starter fluid, that little bit that I added in at the end. For the constant, what was kept the same between all groups? So basically, the kombucha is being kept in a warm and dark place, like the closet that I showed you. The independent variable, so what changed between group to group, and that would be the actual SCOBY itself. You might be changing the type of tea you're using, and you might be using a different type of juice than what I used. And then the dependent variable, what is being measured, what is being recorded, basically what is our final product, and that would be the kombucha drink. So again, I hope that you were able to see how cellular respiration is taking place in this experiment, in this activity. I hope that also maybe you're interested in trying the kombucha drink. That is up to you again. But at this point, your entire lab notebook should be filled out. So please make sure you scroll through, make sure you have answered all of the questions that you need to answer. But all I want to say is great job. We're all done.